Welcome, everyone. Thank you for joining us today. I'm Kibra, a global marketing specialist at IBM Technology, and we are delighted to have you join us. Today, we will delve into the live animal imaging with a specific focus on real-time intravital microscopy and in vivo cellular level imaging in immunology research. Now, let me introduce our presenters. Dr. Pooja Jain is a professor in the Department of Microbiology and Immunology at Drexel University College of Medicine. She also holds a joint appointment in the Department of Neurobiology and Anatomy. And today she will present the live intravital imaging of myeloid cell trafficking into the spinal cord of mice. But before that, we will start with a brief introduction to novel intravital in vivo imaging by Dr. Pilhan Kim. Dr. Kim is currently the CEO and also CTO of IBM Technology, and he is also an associate professor at Korea Advanced Institute of Science and Technology. Throughout the webinar, please feel free to ask any questions using the Q&A box below, and we will ensure to address as many questions as possible. Dr. Kim, please begin with your presentation if you are ready.
Okay, sure. Yeah, so can everyone see my screen?
Yes.
Yes.
Okay, great. So thank you for joining us for today's webinar. My name is Pilhan Kim, and then today, I will try to introduce our technology of real-time intravital microscopy for in vivo cellular imaging of various internal organs in a live animal. Okay, so as many of you may know that the IntraVital Microscopy is a very useful technique that can provide a direct, detailed live video in single cellular level in a live animal model. So in this example, in the middle, this is the time-lapse video obtained from the lung of a sepsis mouse model, and then in green, it's showing the blood circulation, which is fluorescently labeled by intravenously injected FIPC spectrum. And then in red color is actually intravascular neutrophil inside this vessel in the lung. These neutrophils are labeled by intravenously injecting LI6G antibody conjugate with the lead prophyl. And then that antibody conjugate circulate the blood flow and then blood vessels and then label all the neutrophil inside the vessel in red color, as you can see in this video. And then in this video, let me move on to the next slide. Okay, so this is how we manage the lung motion for our intravital imaging. So in the left side, what you're looking at is the mouse model is prepared for lung imaging. In the middle, this is our lung imaging window chamber. So you can see this small suction hole in here. So we can suck out the air from this chamber and then build up the negative air pressure enough to stabilize the motion of this lung against the transparent cover glass like this. And then after this preparation, we can take this 10-minute time-lapse video from the lung of a sepsis mouse model. And then as you can see, suddenly this several neutrophil make a cluster, and then it completely blocked the arteriole here, and then disturb the blood flow in this particular area of the lung. So this is what's really happening in cellular level in the acute lung injury mouse model of sepsis. Okay, let me move on to another example. So, again, this intravital microscopy is a very useful tool to obtain the cellular level imaging of live tissue in a live mouse model. So this is the preparation for the imaging of the popliteal lymph node behind the knee, as you can see in the middle in this photo. And then this is one of the images we obtained in a low magnification. So in this example, this is in green color is actually vessel. So we labeled the vascular endocellular cell by injecting anti-CD31 antibody that can systemically label all the endocellular cell. in this mouse model. And then we inject the red color, red fluorescent T cell intravenously through the tail vein. And then this image was obtained at 1 hour after this T cell injection. And then as you can see, this numerous red fluorescent T cell home to the lymph node, this particular pathogenic lymph node, but they are mainly still stay in the certain area inside the lymph node, which is called HB, high intracellular value. So these arrows, point out the high intracellular value inside the pathogenic lymph node. And as you can see, most of this T cell is still, remain around this high intracellular value at 1 hour after the injection. So if I zoom in, so this is another time-lapse video obtained from this setting. So in this example, this red color is the endocellular cell of the high intracellular value, blue upside. And blue color is the fibroblast reticular cell, again labeled by the antibody conjugate. And then green color in this video, green color is the T cell. So as you can see, the intravenously injected T cell arrives here and then attached to the endocellular cell. And then it quickly, transmigrate through the endocellular cell junction like this within, just 5 minutes. So this is the first step of the T cell, homing to the lymph node. And then if I extend the time period to 2 hours, now we can see the whole process of a T cell, and in this particular example, T cell and B cell extravasation in the this HB of the lymph node. So as you can see, this this lymphocytes, T cell and B cell, they actively infiltrate to the lymph node parenchyma through the certain, endocellular cell junction inside this in this high intracellular value. Okay, so and then not only the cell, we can also image the drug as well, as long as it's after fluorescence labeling. Okay, I'm sorry whether you can see the video here. So this is the real-time video obtained from the liver of the mouse model on this left photo. And in this particular example, we use type 2 GFP mouse model. So all the vascular endocellular cell express GFP. So all this green color is the liver sinusoid endocellular cell in the liver. And then we inject the nanoparticle, the red fluorescent nanoparticles through the tail vein. And then, so when this movie starts again, this is when we inject the nanoparticle. And then after around 10 seconds, this fluorescently labeled nanoparticle flows through the liver sinusoid vessel. And then they quickly, accumulate to the certain area inside the liver, in the liver sinusoid, as you can see in this video. And then we can, easily distinguish the, single flowing nanoparticle inside the vessel. Okay. So now we can extend it actually, the time period in a time-wise, we can extend our imaging time. So this is another example of the imaging of nanoparticle delivery to the tumor model, to the cancer model by using this dosal skin pulse chamber. So this is 2 hour, 6 hour, and 24 hours after the intravenous injection of this fluorescent nanoparticle through the tail vein again. And here, the green color is the triple negative breast cancer cell line, MDMB231. This cell line this cell is implanted inside this dosal skin pulse chamber in this photo. And then again, the blood vessel, this tumor blood vessel, is labeled by the injection of anti-CD31 antibody. And then let me remove the green color here. So at 2 hours, we don't see many nanoparticles inside the tumor vessel. At 6 hours, now we can see the increase of the nanoparticle, but they mostly stay inside the vessel. But finally, in 24 hours, we can see even more increase in nanoparticle, and then many of these nanoparticles, got out of the blood vessel and delivered to the tumor cell. For example, here, here, and also here. So the IntraVital microscopy is a very useful technique to visualize the delivery of the drug to the target cell in a live mouse model, like this. Okay, so this is the short summary. So IntraVital microscopy, they can enable a dynamic 3D imaging of various cellular level dynamics, as shown in this video, in many different organs. And then, we can see, various cellular level dynamics, such as cell trafficking, cell-to-cell interactions like this, or cell-to-microenvironment interaction instead of live, animal model in vivo. And then, as I showed you before in these two examples, with the drug development, the IntraVital microscopy is also, again, a useful technique that can enable direct imaging analysis of drug delivery to the target tissue and cell. And then also we can monitor the drug efficacy in the target cell after the delivery and then validate the mode of action in a live mouse model in vivo. Okay, so our IntraVital microscopy have been used to image, applied to almost all the internal organs in the mouse model. All of these images obtained from the live anesthetized mouse model by using our IntraVital microscopy setup. So as you can see, we have been able to image bone marrow, brain tumor, retina in the eye, skin, hair follicle in the skin, various tumor model, pancreas, spleen, GI tracts, adipose tissue, prostate, placenta in a pregnant mouse model, memory tissue, lymph node, kidney, muscle, heart, lung, and so on. Okay, so let me briefly introduce our dual mode IntraVital confocal and two-photon microscope model, which is called IBM CMS3. So it's a single box packaged, all-in-one packaged system for optimized for in vivo cellular imaging of a live animal model, as the example I showed you before. And then it can do both of two-photon imaging with 4 different colors by using a compact femtosecond pulse laser integrated inside this box. And then at the same time, it can also do confocal imaging with 4 different colors by using whole wavelengths, confocal laser module. And then in addition, this box has additional optical ports on the back side to integrate additional laser systems, such as tunable, more conventional tunable titanium sapphire femtosecond pulse laser system for two-photon imaging. Okay, let me show the short video about this microscope. So as I told you, this microscope can connect to the other additional laser system. And then in the front, we can open up our box. And then we have animal stage. They can place the animal model for imaging like this. And then, by using this in a single box packaging, you don't need the, complete dark room. You don't need a dark room. And then this microscope also can be installed in a very small, space. And then inside, we have, multiple optical components to perform the four-color confocal imaging, and then also four-color two-photon imaging as well. We have two-photon lasers inside our box. And then, for two-photon, we have a four-color, NDD two-photon detector as well. So we can do, simultaneous four-color imaging for both of confocal and two-photon imaging. Oh, sorry. And then, well, many of you already know, the confocal microscope is a very useful technique that can, provide the, section G-stack imaging in a 6 sample, like a live tissue in a live mouse model. And then this is the one representative data obtained from the immunoadipose tissue of the, again, anesthetized mouse model in vivo. Green color is the adipocyte, and red color is all other cell types like vascular endothelial cell and the macrophages. And then as you can see, we can have a very high resolution JSTAG imaging in the live mouse model by using our confocal imaging mode. And then later, we can make it as a 3D rendered data using this JSTAG imaging data. Well, two-photon microscope is another technique. It uses longer wavelengths, near-infrared femtosecond pulse laser, so it can penetrate more. And then it can also do presence imaging, like the confocal imaging, like this. In addition, this two-photon microscope can detect SHG signal, the second harmonic generation signal, generated from the collagen and the other regular protein fibers in the mouse model without any labeling, without any additional labeling. So this is a label-free technique, imaging technique. And then since this SHG signal, second harmonic generation signal, is very efficiently generated from the collagen fiber bundle. So it can be a very useful, signal to visualize the fibrosis in a mouse model like this. So this is the one example obtained from the liver fibrosis mouse model. So all this color, all this green color is the fibrous collagen accumulated in the liver of this liver fibrosis mouse model. So, let me show you just another, four color, simultaneous four color, imaging, two-photon imaging result obtained from the skin by using our pentasecond laser system, laser module inside this microscope. So this is the GSTAC data obtained from the skin. And then green color is GFP, H2B GFP, so from the nuclei. Red color is the ROSA26 membrane pyritomator from all other cells. And then this white color is the collagen, visualized by the second harmonic signal. So as you can see, we can nicely see the fluorescence signal from the nuclei in the cell membrane, and together with the collagen in the skin. So most of these are the dermal collagen in the skin. And then this is again, simultaneous fluorescence and second harmonic generation imaging from the skeletal muscle. And the blue color is actually blood vessel, labeled by the intravenously injected advanced group. So, well, we can do this four-color imaging almost every organ and tissue in a live mouse model. Like this is data from the pancreas. This is another example obtained from the kidney. So we can nicely distinguish the individual tubule in the kidney by using our two-photon microscope. And then, all this data can be visualized in 3D and also in 4D. So this is 3D data obtained at different time point, and then we can, generate this 4D data set. Okay, and then this microscope, another unique feature of this, two-photon microscope is it's equipped is a high-speed laser scanner for real-time imaging. So with 512 by 512 pixels per frame, we can obtain 30 frames per second at default mode, and then you can increase the speed up to 100 frames per second, 100 frames per second. That's options. So by using a rotating polygon mirror, I don't want to go into too much detail, but by using this unique, rotating polygon mirror scanner, we can achieve this very high speed. So it enables us to, directly monitor the flowing cell in the blood vessel. So this is the example, another example obtained from this tumor mouse model. So blue color is the pseudocolor tumor cell, and the green color is the red blood cell. We fluorescent red blood cell. We intravenously inject into this mouse model. So you can see this flowing RBCs in this tumor mouse model, and then we can directly compare how the blood flow is different in this mouse model. And then this is another real-time data obtained from the liver. So again, red color is the RBC and green color is the vessel. So we can nicely see the flowing RBC in the lung. And then we can also do the same thing, similar thing in the heart as well, by using our stabilized heart imaging chamber. And then, our, very recent work, in press in Nature Method is AI processing model for denoising of this real-time video. So on the right-hand side is the raw video in real time. And then by using our new AI processing technique, we can very efficiently they very effectively denoise, remove the noise from this noise real-time video like this. So this is another example, the raw video, and then denoise the image video. And then you can nicely see this growing in a red blood cell in this vessel obtained from the muscle. Another example obtained from the kidney. Okay, and then this high-speed laser scanner, also enables us for automatic live tissue motion compensation function. So, we can, directly monitor, capture all the tissue motion in this mouse model. So, and then while we are doing it, while we are obtaining this live video, we can do frame by frame, image registration to compensate the tissue motion in a live mouse model, like this. So, this motion compensation function is very essential. It's indispensable to obtain this high resolution, the high quality motion stabilized imaging from the live mouse tissue, as I show in this slide. Another example. So this is actually the video obtained from our real-time video obtained from the kidney, and then this is quite common movement we can observe from the internal organs. And then again, you can see the impact of the motion compensation with and without. And then we implemented this function, motion compensation function, to be easy to use. So it can be done automatically. So what you need to do is just turn on the automatic compensation functions. And then we can, from this real-time video, we can automatically generate this motion compensated high excellent images. And then also inside this box, we have taken care of body temperature control by using this tablet PC. And it is tablet controller, and then we can also have this heating and then controlling temperature control system to maintain the temperature inside the box. Okay, so by using this microscope, we have been able to image the almost every organs in a live and excellent mouse model. Then if you're interested, please visit our website, www.ivimtech.com. And then it has information what you can do with the different disease model. And then we also have various webinar series focusing on each specific application like peptide imaging or exosome imaging, for example. And then we also provide the imaging service as well. So if you're interested, you can just send us your material to test, and then we can do all the IntraVital imaging experiment for you, and then give you the data, and then provide you the data. This is one example. So this is the exosome imaging, exosome delivery imaging, 10-minute and 30-minute after the injection. You can see this radical exosome was attached to the delivered to the surface of the target cell, and then later it internalized as 30 minutes after the injections. Okay, so IBM technology, we are providing the microscope, and then we also provide the imaging service as well. And then with our in-house specialist team with more than 10 years of the experience, we can provide very specialized application support. And also we can provide the technical and training service as well. So thank you very much for your attention.
Very well.
Wonderful.
Yeah,
All right. So let me just give you an idea about what we are and how live in neuroimaging has really helped our research, over the years. So my lab is in Philadelphia, Drexel Med School, and our overall goal is to really actually understand how immune system responds to inflammation and infection in central nervous system. So we use this technology for studying, the problems in brain, various types of the diseases. And also when Pilhan was mentioning exosomes, we are working on exosomes, although I have not covered that, so that's another thing that we would be extremely interested in. And I have several collaborators in Jefferson, Temple, and John Hopkins, whom I would be happy to share about this, newer system that you guys have. So just to give you an idea what we are. So for last over 2 decades, my lab has made some seminal contribution and discoveries in the field of neuroinflammation and also virus-induced cancers. So we have discovered a myeloid cell-based target for multiple sclerosis and other neuroinflammatory diseases, and we have done the preclinical validation. And these studies were really started from my introduction with IBM technology. We also have discovered a very new and unique reservoir for HIV, which is in AIDS patient undergoing therapy. And this reservoir is by cervical lymph node. So that also was the project that initiated by my interaction with the IntraVital live imaging. Currently, we are working on a very new immunotherapy strategy that involves neoantigen discovery and immune checkpoint blockade, and we are applying this in the setting of both cancer and neuroinflammation. Finally, we have discovered a new target called MEF2 against a non-Hodgkin lymphoma, but that is something not necessarily related with IBM. So I'm going to just share highlights from our neuro projects. So why it's important to study the brain-body interactions in health diseases, because as the healthcare industry is evolving, we have people living very long, healthy life, but with aging comes the chronic diseases. So one of those chronic diseases are diseases of the brain, and they could be considered as the 21st century epidemics. And these diseases are facilitated by a crosstalk between the brain and our immune system. Because now we know that our central nervous system, including both brain and spinal cord, is capable of mounting dynamic immune responses against infection, trauma, stroke, and toxin. And collectively, we call them NI responses or neuroinflammatory responses, which are associated with several diseases such as multiple sclerosis, Alzheimer, Parkinson, Huntington's, as well as HIV and other virus-associated neurological complications. So this review from Michelle Schwartz from Weizmann Institute really nicely lay out the program for us, that how the most of the problem in our in the brain and spinal cord is because of the failure of brain immune crosstalk. So that will be a suggested reading for those who are interested. So how this really happened? So the human central nervous system is a highly vascularized unit. If you take all the blood vessel that supplies fuel to our brain, you can make over 100,000 miles of blood vessels. And through this intense network of blood vessels, there are two areas called meninges and perivascular curve, where blood-brain barrier is much thinner, allowing lot of opportunity for immune cells to interact with the CNS resident cells, and together they make a neurovascular unit. So neurovascular unit is made up of a highly specialized microvascular endothelial cells, which actually make high endothelial venules within the CNS. And then these are lined by the footpad of astrocytes and pericyte, and also associated with neurons and astrocytes as well. Through the lumen of these microvascular endothelial cells, a slew of immune cells constantly enter into CNS for immune surveillance. One of those cells called dendritic cells, which are most potent antigen presenting cell, have been the focus of our research. Which was greatly facilitated by the consultating advice and collaboration with late Professor Ralph Steinman, who discovered dendritic cell and received Nobel Prize in 2011. So why we interested in dendritic cell? Because they are very strategically located in the three areas of antigen encounter. For example, blood-borne pathogens are sampled through spleen, tissue-borne pathogens are sampled through peripheral lymph node, and the mucosa-borne antigens are sampled through mesenteric lymph node. Now, dendritic cells are positioned in all these three sites, armed by the receptors for protein sensing like toll-like receptor, or RNA sensing by RIG-like, or DNA sensing by DNA-like receptor. So what happens that whenever the body encounters any insult by inflammation or infection or any other pathogen allergic encounter, these circulating dendritic cell capture the antigen and they roll over the endothelials to go to nearest lymph node. Inside the lymph node, dendritic cells are able to process and present antigen to naive CD4, CD8 T cell, and those cells become activated and come to periphery where they are primed to deal with the problem that body has encountered. So pretty much the entire choreography of immune system sets place like that. And this is also the fundamental of vaccine, how the memory response are created. Now, with the 2-photon imaging, we can very nicely see, so this is also coming from the similar to what film showed, imaging the lymph node of animal for the localization of T cell area and B cell area, and superimposing with the circulating dendritic cell. So as you can see, the macrophages are pretty much on the margin. They are called marginal zone macrophages, and dendritic cells are embedded within T cells and B cell area. So T cell moves on conduits, but B cells move on a very specialized dendritic cell called follicular dendritic cell. So my interaction with the dendritic cell migration is started by my visit to the Theodor Kocher Institute in the University of Bern, Switzerland, where I work with these German scientists, Britta Engelhardt and her team. And we together for the first time provided evidence of the dendritic cell migration into the CNS. So when we were doing it, so we had to go with the conventional method where we obviously start with the area prep like film showed, associated with a high sensitivity video camera, and then collecting image and processing. Now, when I was interested to see the behavior of dendritic cell in brain, I could use 2-photon microscopy, because 2-photon microscopy requires the area under view should be very stable. So this is again film. This image is like 2006 or 7, very early first generation 2-photon microscope. So what we did is we created a meningeal window in the skull of mouse, which is normal, and we injected 2 quantum dots to expose the meningeal vasculature, and then labeled our immune cells, with CFSC, and introduced IV. So what we notice that when the CNS is very intact, there are few cell cross blood-brain barrier, but when there is no problem in the brain, no inflammation, they come back. So we showed that why people thought that CNS has no immunity, because these cells undergo constant surveillance, but they come back in the periphery. Now, what happened in the inflammation? So, for inflammation, the best model is EAE, experimental autoimmune encephalomyelitis, which is caused by the infiltration of immune cell in CNS, and it's a good model to study multiple sclerosis. So we use 2 type of EAE model, and then we did our preparation. So this requires 2 type of preparation. Here, what we did is a microsurgery on the heart and introduce a catheter into the carotid artery. So carotid artery feeds into the CNS directly. And then we expose the spinal cord and put a film and to expose the entire spinal cord. So remember, these animals are not naive. They are under high inflammation. They have hind leg paralysis. And second thing is that because we have to image the entire spinal cord, we could not use the 2-photon microscope, because the area under observation is under highly mobile. So this is a video microscopy. So that is something for you guys to look at it for the larger area. How can you really use the 2-photon? So we use the video microscopy, because the spinal cord is under constant movement, and we cannot stabilize, and we wanted to see the behavior of cells throughout the spinal cord. So here we are injecting our dendritic cell through IV in the heart and observing their entry into CNS. So as you can see, because the microvasculature is highly inflamed, thousands and thousands of myeloid cells cross the blood-brain barrier and they enter into the CNS, making very firm contact. So this was the first evidence provided that what happened in the people who are going through constant inflammation, there is a burst of immune cells entry into the CNS, and these cells, like we show in normal animal, do not come back in circulation. They make a very firm contact. Then finally, when animals survived 2 hours after surgery, so we could find where all the cells went. So we showed that a lot of cells are accumulated into meninges, where I showed you that it's a thinner blood-brain barrier, but we could find cells in parenchymal, perivascular, and intravascular regions. So the cells enter through various regions into the CNS. Then when I came back from Switzerland, I collaborated with a lot of people in the radiation oncology team in Johns Hopkins, and we showed that this is a near-infrared imaging. So in near-infrared imaging, you can image the entire, it is still live imaging, but it's non-invasive. So you can image the entire CNS, including brain and spinal cord, and observe the behavior of immune cells trafficking. So here, using the probe label antibody, we are showing that as the degree of inflammation increases in 3 different animals, the migration of myeloid cells pattern increases. So by the animal, when it's fully inflamed by score of 3.0, the dendritic cells who were limited to the half the length of spinal cord reach up to the brain, and they co-localize with T cells and myelin basic protein. It means they are able to do in situ antigen presentation and setting up this cascade of neuroinflammation. Followed by that, we provided evidence in the 3LL cancer model, where we introduce our PSMA labeled disease in the close to footpad, and then we noticed that if the tumor was spread in the brain, the cells over the period of time develop the reach to the area of tumor site. Similarly, using echo MRI, we showed that if you have a tumor introduced in one site of rat brain and you put the radio labeled dendritic cells, over the period of time they move to it. So pretty much by various imaging tools, we were able to provide that lot of these cascade of neuroinflammation is caused by the crossing of cells to the blood-brain barrier and their constant entry into the CNS. And these studies we able to apply in the inflammation model, such as MSEAE, in the tumor glioblastoma model, and infection, HIV, and neuro-AIDS. Now, given with respect to the time, I'm not going to go through all the data, but I will just provide you the main headlines for the MS. So all our MS related work has been published in Nature Scientific Report, where we showed that one of the single dose antibody treatment of CLAC12A can attenuate the disease in animal and provide protected immunity. Now, this paper has both data from in vivo imaging as well as lot of functional studies. Now, followed by our study, the paper in Nature 2015 by Jonathan Kipnis' lab actually showed, again by the live in vivo imaging, that there is a very functional meningeal lymphatic system that runs across the skull of a mouse, and it has high concentration with the sinus area. And what they showed, that there is not just a migration of cells from periphery to CNS, there is a retrograde transport of the cells that enter into the CNS through this meningeal lymphatics, and they lead into drain into cervical lymph node. So this is the another another cartoon created to show what happened when the lots of immune cells enter into CNS upon inflammation. They gain access to the rostral migratory system from basal ganglia, and then they travel through the newly generated neuroblast. So the neuroblast, which are constantly produced, they come back into the peripheral circulation. So any immune cell, T cell, dendritic cell, neutrophils that enter, they migrate through these neuroblasts, and they find the home in cervical lymph node through nasal mucosal lymphatics. So using this information, we actually were able to standardize a very unique reservoir for HIV. What we showed, again, all this data is published, I don't have time. What we showed that dendritic cell, when they take HIV virus, they can sample the virus in CNS and they bring the virus back through rostral migratory system, and they put all the virus which actually was in CNS, they protect that virus in the cervical lymph node on follicular dendritic cell. So this was, we showed using 11 different rhesus macaque, and the provided the evidence of virus reservoir being in this. Again, all this work is published, feel free to look at it, but the point is that imaging studies could be very functional and they can highlight the, what's really happening. So with that, I want to just thank the various students who over the year contributed to the work, and also my collaborator within the United States and across the globe. So with that, I will end and take any question that you guys might have.